Hello, my name is Aaron RV, and I'm going to describe to you some exciting new research regarding the regulation mediated by microRNAs and siRNAs. This work was done in collaboration with Christina Leslie and Deborah Marks at Memorial Sloan Kettering Cancer Center. MicroRNAs and siRNAs are small RNA molecules that are able to mediate degradation and inhibit translation of mRNA transcripts. Thus, microRNAs form a post-transcriptional method for gene regulation. One important problem in the microRNA field has been determining which microRNAs regulate which genes. This has historically been explored by examining regions of complementarity between microRNAs and 3' untranslated regions of genes. While this provides a discretized prediction of either a target versus a non-target, we believe that downregulation is more of a continuum. Furthermore, we believe that there are systems-level factors that quantitatively influence microRNA-mediated downregulation. Specifically, we find that target mRNA transcript abundance can limit microRNA-mediated downregulation. For instance, here we see on the left two microRNAs, microRNA A and microRNA B. MicroRNA A has very few targets, whereas B has many targets. In this case, we might expect that A would be able to degrade all 3 out of 3 of its target transcripts, whereas B would only be able to degrade 3 out of 12 of its target transcripts. Thus, A's targets would be functionally irrelevant, whereas B's targets would still be translated into functional proteins. On the right, we see a schematic of this hypothesis when there are many different microRNAs with varying concentrations of target transcripts. On the y-axis is mean downregulation of microRNA target, and on the x-axis is total target concentration. Our hypothesis is that targets will be less downregulated as target concentration increases. When we performed a meta-analysis of 143 publicly available datasets, we found that this phenomenon empirically occurred in human HeLa cells. We determined the downregulation mediated by microRNA by transfecting the microRNA into the HeLa cells. We then measured change in gene expression by microarray assay, and we determined total target concentration by high-throughput RNA sequencing, units of which are reads per nucleotide, or RPN. On the left, we see mean downregulation by a microRNA, as shown on the y-axis, is highly correlated with target concentration, as shown on the x-axis. We see that miR-142-3P, which is the microRNA with fewest number of targets, is able to downregulate its targets nearly twice as much as miR-16, which is the microRNA with the most targets. Since siRNAs have been shown to act like microRNAs on off-targets, we examined the mean downregulation of off-targets of siRNAs. On the right, we show that there is again a highly significant correlation. Thus, if it is not feasible to design siRNAs with very few off-targets, it may be preferable to have many off-targets such that each individual off-target is minimally downregulated. However, we also show that excessive number of off-targets can decrease the impact on primary target downregulation. We examined the primary targets of siRNAs to determine if they were affected by off-target abundance. Here we see that 4 primary targets respond differently when knocked down by siRNAs with varying nucleic acid sequence, and thus varying numbers of off-targets. When we combine all these experiments, we see a highly significant correlation between the downregulation of the primary target and off-target abundance. Thus, siRNA-based drugs designed to have few off-targets will be more effective in downregulating their primary target. We also did a more controlled analysis where we examined individual genes that are regulated by more than one microRNA. As shown here, the downregulation of shared targets, which is the same for both microRNAs, is dependent on the concentration of non-shared targets, which is specific for each microRNA. Example genes that are highly differentially regulated are shown on the left. On the right, we take the average difference in downregulation and the difference in total target concentration to find that they are highly correlated. Better understanding target concentration of their systems-level factors will shed insight into several critical problems, including better understanding varying levels of regulation by microRNAs, the effects and artifacts of microRNA transfection experiments, optimizing siRNA functional screens, and designing siRNA therapeutics. Thank you for your attention. If you have any questions or comments, you can reach us by email or visit our poster at the upcoming Cold Spring Harbor Systems Biology Conference starting on March 23rd. Otherwise, we'll be seeing you at ISMB in July.